Okay, so a little bit about DNA, genes, chromosomes. So, what makes you who you are from a sort of structural point of view is all down to your genes. And your genes are like little instructions that are written instead of our alphabet with 26 letters. There's just 4 letters in the genetic code, and they are A, T, C, and G. And the order of those letters determines what proteins are made and how you're made. And this is the sort of the famous double helix, and that's folded up tightly, tighter and tighter and tighter and tighter and tighter, and then it's packed into these things called chromosomes, and those chromosomes live in the middle of the cell called the nucleus. And nearly all of the cells in your body have got one copy well, they've got 2 copies, I'll come on to that, 2 copies of all of your genes, but they've got one package of your genetic material in each cell of your body. Okay, so chromosomes are these like sausagey-like things that carry all of your genes, and the particular chromosome where the nail patella gene lives is chromosome number 9. And you can see that they come in pairs because you get one from your mum and one from your dad. And the numbers are just there to tell you whether they're sort of long ones or short ones. So clever people in the labs look at them down the microscope and they can tell from the stripy pattern whether it's a number 1 or a number 2, all the way down to a number 22. And then if you're a boy, you have an X and a Y, and if you're a girl, you have 2 X's. And all of your genes are all jumbled up on each chromosome. So it isn't like chromosome number 9 is for your legs and chromosome number 10 is for your brain, that type of thing. They're all jumbled up. And this was just a little sort of genetic artwork made out of socks, actually. So if you can see close up, these are actually all stripy socks made to look like chromosomes.
I lost that so last week in the washing machine.
Okay, so what do genes do? Well, so as I say, they're like instructions or little recipes in a great big recipe book. And each gene is an instruction to make a protein. And proteins make the structure of the body and make the body function properly. So structural proteins are like collagen, that's an example of a structural protein, and a protein that makes the body function properly is like insulin. But some proteins don't do anything directly themselves structurally, but they control loads of other genes. So this is particularly important in nail patella because this is one of the master controller genes. So some people think, well, how can one gene cause problems with my eyes, my kidneys, my bones, my nerves, all of those things? But that's because it's what we call a transcription factor or a master controller. So it doesn't just do one job, but it actually controls the way loads of other different genes in different places work. So that's why you have lots of different problems with one condition caused by one gene. And as I say, all of your genes come in pairs, because you get one from your mum, who doesn't have to be a ballet dancer, and one from your dad, who doesn't have to play baseball. So you get 2 copies of everything. And altogether, we've got about 23,000 genes, and I've just had to change this from 30,000. As more and more is known about the human genome, the number of genes that we've got actually keeps on coming down. And when I started in genetics, we used to say we've got about 100,000 genes, so now it's about 23,000. And the gene for nail patella syndrome is called LMX1B, and LMX1B is found on chromosome number 9, and the particular bit of chromosome number 9 is called 9q34. And if you remember the stripy pattern on the chromosomes, those stripes have got little numbers, and Q means it's the bottom bit of the chromosome and P would be the top bit. So that's all the technical gear, and I'm just telling you that in case you read it somewhere and think, what does all that mean? So how do you actually get nail patella syndrome? So if you remember that all of your genes come in pairs, to have nail patella syndrome, one copy of the nail patella gene, the LMX1B gene, has to have a spelling mistake in, a fault in. And the technical term for a spelling mistake or a genetic fault is a mutation. I don't sort of like the word mutation because it just sounds a bit, you know, horrible. But the technical term is a mutation. So if you've got Patella syndrome, you will have one copy with a spelling mistake in, and the other copy will be a normal copy that's working properly. And the one with the spelling mistake in is dominant over the normal copy. So you might hear about dominant inheritance, and that's what it means. It's the faulty copy is dominant over the normal copy, and you get the condition. And the important thing to know is that we all actually carry about 5 faulty genes, but most of them are hidden away, and you need 2 faulty copies for a lot of conditions like hemochromatosis, cystic fibrosis, and that. So we're all carriers of conditions we don't know about, but for those conditions you need 2 faulty copies. With Patella syndrome, you just need the 1 faulty copy to be dominant, and then you get the condition.
My question is, you turn around and say about 2 copies there. Is it true that we have actually got a good copy of NMX1B in us?
You have.
Even though we're affected?
You have.
Can't be, because I remember Ian McIntosh turned around and saying in his video, with 2 people who had NPS, just while I got my head around it, if 2 people who had NPS had kids, the chances of their kids had it went down from 50 to 25%.
Yeah, so
we do actually have 1 good copy.
That's right. I will come on to that in a bit, and I'll tell you about that. So if I don't cover it, remind me, and I'll come back to it. So you're absolutely right. So because all of your genes come in pairs, so you will have 1 normal copy and 1 copy with the spelling mistake in. So how do genes go wrong? My genome made me do it. So as I say, they're like instruction books. And everyone with Patella syndrome will have 1 spelling mistake. The important thing to realise is sometimes you go to conferences and people say, well, what's your mutation called and what's your mutation called? Everybody with Patella syndrome will probably have a different spelling mistake to another family with Patella syndrome, but that doesn't really matter. The fact is it stops the Patella gene working properly, and that's enough to give you NPS. So it doesn't matter what type of spelling mistake you've got. That doesn't predict whether you're going to have kidney problems or eye problems or anything like that. But within your family, you will all have the same spelling mistake because it's that spelling mistake that's passed down through the family. So different families will have different spelling mistakes, but all in the same gene, all in the Patella gene. Does that make sense?
Does that mean that if you have, if it's passed down within a family, that you would expect the same symptoms to show?
No.
Right.
So if you remember at the very beginning we were talking about variability, it's incredibly variable, not just between families, but within the same family. So a lot of the time the diagnosis is only made in a family when a child is born with a really obvious problem, bad feet, bad knees, that type of thing, but then you look back and say, like, one lady only was diagnosed with Patella syndrome in her 70s when her grandson was born with bilateral club foot. And she said, oh, I just thought they were my thumbnails. And she'd gone through a life with no orthopaedic problems, no kidney problems, and they thought it was just their family thumbs. And she was still doing great big high kicks, which she demonstrated to me in her lounge in her 70s. So it's really variable within the same family. So the spelling mistake doesn't predict the way it's going to have an effect on you. And members of the same family will share the same gene change. So spelling mistakes are mutations, and the way those mutations happen is you can have, if you think about this code again, you can get some letters swapped for another one. So if you imagine in normal English, if you swap a letter with another letter, it completely changes the sense of that word and can change the whole meaning of the sentence. So you can either get letters swapped for another, and they're called missense mutations, just in case you read any of this and wonder what it means. Or you can actually have letters missing or extra, or letters can change to a stop signal, so you can actually stop a protein being made, or you can have whole chapters of a book missing rather than just letters. So there's lots of different ways that genes can go wrong, and there's lots of technical terms for those: nonsense, frame shift. Some people are maybe more interested in this than others. I know some people do read technical articles. So nail patella syndrome, as I say, is caused by this LMX1B, and it's a really interesting gene. As I say, it's a controller protein, and it's mainly important when the embryo is actually developing in the body, because it's expressed, it's working in particular places in the body at particular times of development, but it's working in the kidneys all the time. What does it do? Well, one of its master controller functions is to do with patterning the body and telling the body whether to be, with bits of the body to be the front of the body or the back of the body, and I'll show you some examples of that in a minute. It's also important in the kidney and the eye and the brain and the nerves. So these are little mice. Now, believe it or not, you can have nail patella mice. You have to give them nail patella by some genetic manipulation. This is work that I haven't been involved with, but was done in Johns Hopkins. Now, if mice have just one faulty copy of the nail patella gene, they actually don't have any problems. So to give them nail patella syndrome, you have to give the mice 2 faulty copies. So they're a little bit different from humans with nail patella. But this is just to show you where nail patella affects the mice and where the gene is expressed, so where the gene is active in the developing embryo. So the black bits are where the nail patella gene is being expressed. So you can see this is as the embryo gets older. You can see here it's the area that goes on to become the brain. Here, these are the little developing limb buds. You can see in the developing spine, and you can see in the developing eye. Can you see? So as it gets sort of older.
Please tell me that's a tail.
Pardon?
Please tell me that's a tail.
Yeah, yeah, you're all right. That is a tail. And here, can you see in the kidney? So you can see the nail patella gene being expressed in the limbs, in the kidney, in the spine, and in the brain. Okay, so if you remember, I was talking about telling the body to be whether to be the front or the back. Now, various genes are important in patterning. So they're important in making your arms grow outwards, making this side of the hand become a thumb rather than a finger, and making this side of the hand become the back of your hand rather than the palm of your hand. And if you're missing certain signals, various things don't form properly. So actually these mice who are completely missing the nail patella gene have normal palms of the hand. So this is normal, that's called wild type. So you can see here a normal mouse, this is the back of its paw, this is the palm of its paw, and that's side on. But a nail patella mouse without the right signals from the nail patella gene, this is the back of its hand, which looks completely the same, almost completely the same, as the palm of its hand. So you can see that nail patella is really important as a signal to tell the developing hand or paw to become the back of its hand. So that's called dorsoventral patterning. So it doesn't pattern properly.
How does that translate into humans?
I'll tell you.
Oh,
So you can see, obviously it's not directly comparable with a mouse to a human, but you know the swan necking that some of you can do? That's to do with the tendons at the back of the hand not being formed properly. So some of the features of nail patella are due to structures which are on the back of the body not forming properly. Now you can see that this is the back of your hand, but you might say, well, hold on, my kneecaps around the front, so how does that work? Well, actually, if you think back to how embryos develop, when you're developing in the womb, if you imagine, you know the way how sort of crocodiles walk, they sort of walk with their kneecaps, if they've got kneecaps, but their knees and their elbows towards the back. And actually, in the womb, the knees and elbows are what we call dorsal structures, so they're at the back, but you get the legs rotating round to the front later on during the pregnancy. So from an embryological point of view, your kneecaps are actually structures associated with the back of your body. So a lot of the orthopaedic problems are due to the back bits of you not forming properly because they haven't got the nail patella signal to tell the back bits of the body to form properly. That's incredibly, you know, simplifying things, but that's the sort of basics. So how is it inherited? So I mentioned the word autosomal dominant before. Some of you might have been to genetics and heard of that word. So it means that the faulty copy is dominant over the normal copy. So how does that work? So if you've got somebody with nail patella syndrome, they've got one faulty copy and one normal copy. And each time you have a child, you pass on just one copy of all of your genes. And which one gets passed on is just random. It's just like tossing a coin. So each time you have a child, and it doesn't matter what you've had before, because like chance doesn't have a memory, so it doesn't matter if you've got 3 previously affected children or no previously affected children. If you've got nail patella syndrome, you would have a 50-50 chance of either passing this faulty copy on or this normal copy. And as long as the unaffected parent, as long as the other parent doesn't have nail patella syndrome, it doesn't matter which copy of their LMX1B gene that gets passed on, that will be normal. So basically, if you've got nail patella syndrome, you've got a 50-50, so a 50-50 chance of passing it on to a child. Now, your question about what happens if 2 parents have it.
Oh no, it's just because I think it's getting my head around that.
Yeah, so does that make sense from
yeah,
the, so that's the usual situation. Now, it's very unlikely that 2 people with nail patella syndrome would meet each other apart from at a conference. So
each time it's about the 25%.
Yeah,
one another word.
yeah. So if 2 people did come together and have children, instead of there being 2 options, there would be 4 options. And one of those 4 options would be for the child to have 2 faulty copies. And to my knowledge, that hasn't happened to parents coming together with nail patella syndrome. But if it did, there would be a 1 in 4 chance that they would have 2 faulty copies, and we would predict that actually the baby wouldn't be able to survive. So, and as I say, so it's a 50-50 chance for each child. Doesn't matter what you've had before. It's just like tossing a coin. It doesn't matter if the baby's a boy or a girl, and it doesn't matter whether it's the mum passing it on or the dad passing it on. It's still a 50-50 chance. And it's really important to remember that it might be milder or more severe than the parent, and it's really unpredictable. So the 50-50 chance is the chance of passing the gene on, but it's not the chance of passing kidney problems on or knee problems. That's just the chance of passing the gene on, and you can't predict how that gene will have an effect. So this is a picture of dominant inheritance. So you can see that this person's had circles are girls, squares are boys, and there's a 50-50 chance. So this person here happens to have had 2 unaffected children and 1 affected child. And if you've got it, you can pass it on. If you haven't got it, you've inherited the normal copy of the gene instead of the faulty copy. Therefore, you haven't got the faulty copy to pass on. So if you haven't got it, you can't pass it on. It doesn't stay hidden away. If you've got the gene, then it shows itself up. So the other thing to say is it doesn't skip generations. So if you've heard of, say, a grandparent having it and a grandchild having it, the parent will also have it, but it just may be really subtle. So it doesn't skip generations. If you don't inherit it, you can't pass it on. And it also doesn't get worse over generations. Some genetic conditions get worse down through the generations. If you notice that in a family, it's more likely to be coincidence.
What's spontaneous?
Okay, so next slide. That's okay. So well timed. I'm glad the slide came in that at that point. Okay, a lot of people say, well, why don't my parents have nail patella syndrome? So in my study, about 20% of people, their parents didn't have it. So they were the first in their family. And, you know, why has that happened? Well, basically, when you make sperm cells or you make egg cells, you've got to copy all of your genes, those 23,000 genes. You've got to copy them each time you make an egg cell and a sperm cell. And it's actually more of a curiosity. Why doesn't it happen more often? Why don't these spelling mistakes happen more often? So it's purely a copying error. When the one egg cell or one sperm cell is made, that gene change happens, goes into the egg cell or goes into the sperm cell, and that child who is a result of that egg or sperm cell, all of their cells will then have that gene mutation, and then they've got it completely, and then they can pass it on. So it has to start somewhere, but it's in the formation of the egg cells or the sperm cells.
So it can happen to anybody then?
It can happen to anybody at any age. So some genetic conditions are more common in older mums. Some conditions are more common in older dads, actually. It can happen in any ethnic group. It's nothing to do with what you do or don't do in pregnancy. Some mums particularly think, well, you know, was there anything I did in a pregnancy that's caused it? Anything I could have done? It can happen to anybody from any background, in any country, at any age.
My mum was always worried because when she was pregnant with me, she took a drug called Primodos.
Right.
Stop sickness, I think,
Okay.
in the 50s, I
Yeah.
suppose.
Yeah.
She was always very concerned about that.
No, so it's a genetic thing, and it's literally just that one egg cell or that one sperm cell. But it's the natural thing to do when you have a child who, you know, is born with a particular difference or abnormality.
Somebody who's guilt-ridden.
Yeah, and it's usually the mums. Yeah, and it's nothing to do with having a drink at a wedding or anything like that. It's just one of those things that can happen to anybody. So each time you have a child, you have to copy all of your genes, put them in the egg cell, put them in the sperm cell, and the spelling mistakes happen then. So some people ask us, well, okay, me and my partner haven't got Patella syndrome, but we've had a child with it. What's the chance of us having another child with it if we haven't got it ourselves? Well, the chances of that are very low. Now, we don't say the chances are zero, or the chances are the same as the general population, because there's a theoretical risk that there could be a little cluster of egg cells or a little cluster of sperm cells that have the gene change in, rather than just the one egg cell or the one sperm cell. So we say that if neither parent has got Patella syndrome, the chances are very low, but they're not zero. So prenatal diagnosis, this is a very personal thing and a personal choice, and some people ask us about it and some people can't believe that people are asking about it, and it really depends on your own personal experience, whether you've got Patella syndrome yourself, what your experience of the condition is, or what your experience of your child's sort of journey with NPS is. So this is really for information rather than recommending people should have prenatal diagnosis. We don't do that. This is just information because people ask us what is possible so that they can then think about it. So some people want a prenatal diagnosis for Patella syndrome. Before going down this route, you've really got to ask yourself, well, why am I doing this? Why am I thinking about having a test during a pregnancy? Because if it actually wouldn't change what you would do in the pregnancy, is it really worth going through that? So what happens, the main sort of test for Patella syndrome, firstly you have to know the gene change that you're looking for, because the test is done looking for the gene change rather than looking for features on a scan. So first of all, they have to know what particular family gene change you've got. So that's best done before pregnancy. So if anybody is thinking about having a prenatal test, it's best to get your gene change discovered. And you know,
They can't actually test it without knowing that.
No, because they have to know, because they sort of have to know what spelling mistake to look for first. So you have to know what your particular spelling mistake.
How hard to find in a newborn? Is it harder to find the gene change in a newborn kid than if you don't know, as compared to a normal NPS person? Because they when you take a normal bit of DNA, obviously they search for the code.
Yeah.
To find it. Is that harder to find in a?
Well, you wouldn't test the baby first, because you wouldn't know. If you didn't find it, you wouldn't know whether that was completely reassuring because you might not be able to find it in the parent. Do you see what I mean?
Right,
so
yeah.
if you know what the parent's got, you know it's definitely there, you know it's definitely discoverable, therefore you would then test the child. That would be the order that you would do it in, if it's what you want. A lot of people aren't bothered at all and can't believe people ask about prenatal diagnosis. So what you would do is you have to know the gene change first, but once you do know, early on in the pregnancy you have a scan to check your dates at about 7 weeks. Then at about 11 weeks you have what's called a chorionic villus sample, or a CVS. As I say, that's about 11 weeks. And that's a needle through the tummy wall under ultrasound control. They take a tiny piece of the placenta away, which is made of the same genes as the baby, and they look for the particular family gene change. The results take about 2 weeks, and there is a risk of having a miscarriage because of having that test, and that risk depends on who's doing it and what their particular figures are, but it's about a 1% chance of having a miscarriage just because of having that test. What it can't do is predict severity. All it can do is say whether that baby has inherited the gene change or not. It can't say whether the baby's going to have kidney problems, eye problems, knee problems. All it does is has it got the gene or not.
Will they monitor you though throughout your pregnancy and give you more scans and stuff to see?
I think so. You wouldn't have to have a prenatal test to have extra monitoring. I think, you know, if you're just worried about that being the only way you would get extra monitoring, you know, I think they should be, you know, taking good...
I did have this done just so that I knew.
Yeah.
And it, you know, luckily it's turned out absolutely fine.
Yeah.
But I would have thought if there was something wrong that they would do more scans and stuff to see how it's developing.
Yeah, it doesn't usually cause much problems for the baby in the womb that would affect the baby in the womb's health, if you see what I mean. So the only sort of thing that there's two things with the pregnancy. Firstly, has the mum got NPS herself? Because if the mum's got NPS herself, then she needs extra care for her kidneys. Yeah. Make sure she doesn't get preeclampsia. Make sure the kidney function's okay. Is the pelvis particularly small? Is there a higher chance of having a caesarean section? So there are those mum issues, if you like. But in terms of the baby's health wouldn't be at risk in the womb, if you see what I mean. So they wouldn't need more monitoring of the baby to make sure nothing goes wrong with the baby, if you see what I mean. So it would only be whether any particular things like clubfoot and things are picked up on scan. So you wouldn't have to have the test in order for the baby to get monitoring, because it wouldn't sort of need monitoring for any particular health issues for it in the womb, if that makes sense. So as I say, the invasive test, the CVS, all that does is say whether it's inherited the gene or not. It can't say how the baby's going to be affected. Sometimes ultrasound scans can pick up some abnormalities, but they usually have to be quite severe abnormalities. It's not going to pick up subtle knee problems, subtle elbow problems, but it might pick up a club foot, sometimes the sort of rotational deformities of the legs. So an ultrasound scan's not going to really tell you whether your baby's affected unless something's obvious. Okay, so some people say, well, actually, I don't want a baby to be affected by Patella syndrome, but I don't want to get pregnant, have a prenatal test, and then have a termination if the baby's affected. So are there any other things you could offer? So there is a technique called pre-implantation genetic diagnosis, or PGD, and this is only offered in a very few centres. I think it's been done once for Patella syndrome in Australia, but I have had some people inquire about it in this country, and it would be available if anybody wanted it. It sounds easy, but it's a big palaver, and it's a big palaver physically, emotionally and financially. It involves lots of trips to hospital. You may get the IVF part of it funded, but you may not get the drugs part of it in terms of stimulating the eggs, the ovaries to produce lots of eggs, and also the diagnosis bit, the genetic diagnosis bit. But if I talk you through it, basically the mum is given fertility drugs to make her ovaries produce lots of eggs. Loads of eggs are produced. They are harvested by with a great big needle, sometimes under sedation, sometimes not. And then the eggs are fertilised to produce embryos. And when they get to the stage where they're about sort of 10 to 16 cells, one of those cells is removed from each embryo and analysed to see whether it carries the faulty gene. Now, at this stage, those cells aren't yet destined to become brain or leg or other tissue, so it doesn't matter whether you take one away or not. All the other cells can make up for that, so you're not removing a bit of the baby at this early stage. So you get a load of eggs, you fertilise them, and they develop into sort of 10, 16 cell stage. You then remove a cell from each. By this method here, you can see that down the microscope, this little ball of cells, one of those cells is removed and taken off to be analysed. And then the idea is to just put back embryos that haven't got the condition. Now, it all sounds, great, where do I sign up type of thing. However, you've got to get enough eggs, but not overproduce them and make the mum unwell. They've got to fertilise, some of them won't fertilise. When they've fertilised, some of them won't divide. When they've divided, they may be dividing abnormally, they may not look healthy down the microscope. When you test them, some of them may not survive the testing. Some of the test results may come back unclear. So it all sounds really easy, but at the end of the day, you may only be left with one or two suitable to put back, and they may or may not take. At the end of the process, the chances of sort of taking a baby home at the end of it are about a third. It's about sort of 30%. And it depends on where you go for it. So some people feel that that's something they want to go through, and other people say actually that's not for me. But it's just to let you know that it's out there and it exists. And I think that is my last slide. Yeah. So the main thank you is to everybody who the lovely people I met in the study, and I've met at all the conferences since. And Brian says that I won't be learning anything, but I learn things all the time when I meet people, and it's a pleasure to sort of work with Patella syndrome and work with everybody. So thank you very much.
Thank you.